Okay, here's the video for biochemistry. I know some of the words are small, so but I'll still talk about them. So hopefully it looks bigger on your computer and you can like zoom in or something, or just looks better full screen. I don't know. But biochemistry. First off, just a few things about it. We'll talk about dehydration, synthesis, and hydrolysis. Breaking down word parts. Synthesis means put together. Hydrolysis, the lysis part, means to break. So dehydration, synthesis, removing water to make a bond. We're going from monomer to polymer. Hydrolysis is the exact opposite, adding water back in to break a bond, going from polymer to monomer. Remember, if you think about polymer as a train, each individual car would be the monomer. So that's just what that is. We talked about this a lot. I'm pretty sure I drilled this into your head. Structure always determines function. So that's it for the basic kind of random facts. Then we'll go into the actual 4 macromolecules. So carbohydrates are the first one. Their monomers are monosaccharides. They provide short-term energy. Typically, their names are going to end with "-ose", like glucose, fructose, maltose, dextrose, cellulose, those types of things. There are a few weird ones like glycogen and starch that don't have it. But typically, those "-ose" names. Proteins, they're made up of amino acids. There's 20 of them. There are too many functions, so we don't usually give them one. Some of their examples are things like keratin, myosin, hemoglobin, actin, those types of things. They have the 4 different levels of structure. So there's primary, which is the sequence of amino acids. Secondary are the alpha helix, so those spirals, and the beta pleated sheets, those little kind of bendy parts. Tertiary is the overall 3D shape, so it either looks like a glob or it could be like a fiber looking thing. And then quaternary, the 4th level, is multiple tertiary levels put together, so it's like when they stuck 2 little parts together. Lipids, their monomer is glycerol and fatty acids. They provide several things. They provide protection, insulation, and long-term energy. They have 5 different classes of the molecules, so fats, oils, waxes, steroids, and phospholipids. I think that part kind of tripped a lot of people up. They left off some of those, so make sure you do know those 5 types. And then the big thing about them is that they are non-polar, so they do not have an overall charge, meaning they do not mix with water. Nucleic acids, their monomer is nucleotides, and it has those 3 parts. It has a sugar, a phosphate, and a nitrogen base. Those bases we talked about, there were 4 of them: adenine, thymine, cytosine, and guanine. They're going to store the genetic code, and the only 2 examples of them are DNA and RNA. Then we also have enzymes, which is a type of protein. This protein, these enzymes, they usually end with ACE. Overall, they speed up reactions, meaning they can make or break bonds. They can be denatured by a couple things: high temperature and wrong pH. Remember, denaturing changes the shape of the enzyme. So if we change the structure, we change the function. So they no longer work. They end up getting denatured. And then here's a drawing of an enzyme. So this is the enzyme. On the enzyme, where the substrate binds is the active site, and then whatever the enzyme is working on is the substrate. This part tripped a lot of people off. They didn't know where to put the active site. Active site is part of the enzyme itself.